Oban. The floor is yours y thank you very much por toda la organización de este evento. Muchísimas gracias. The floor is yours.
Thank you very much, Claudia, for the...
Muchas gracias, Claudia, por esta presentación. Es un placer presentar hoy aquí EU-OPENSCREEN, la infraestructura de investigación europea sobre...
Can I move the slides? I can move the slides, sorry. Here we go. So the mission of EU-OPENSCREEN is to identify and develop chemical compounds which can be further developed into drug leads and candidates, or which can be used as research tools in basic science to probe a cellular protein or therapeutic target. As a distributed international research infrastructure, EU-OPENSCREEN provides access to technologies, for example, compound screening platforms, to screen a large number of compounds and to identify those few compounds that have a specific desired biological activity. And also access to resources like large compound collections of more than 100,000 compounds. So compound collections which are large and diverse enough so that there is actually a chance to find such suitable compounds. Also access to expertise. For example, we have a network of expert centers in chemistry in order to develop these compounds further so that they are useful probes in basic research, or that are interesting enough for biotech companies or larger pharmaceutical companies to take up these projects. We also committed to the verification of data, and we make our data, mostly the bioactivity data, in our database after an optional embargo period of up to 3 years. And importantly, we also offer various training opportunities, mostly webinars these days, but also staff exchanges, practical workshops, or 3 weeks ago, we just organized our biannual scientific conference, the European Chemical Biology Symposium. We are working with scientists from both academia and industry worldwide. But just let us take a minute and look together at the main causes for the high attrition rates in clinical phases 2 and 3, which, as you can see here on the pie charts, is lack of efficacy. And we all agree that there are several reasons, not only one reason underlying this observed lack of efficacy. However, a direct consequence of these numbers is that the pharmaceutical companies reduce their efforts in early drug discovery, or they outsource these activities in early drug discovery, and they focus on those very few targets that have been thoroughly validated by using chemical probes with a well-defined mode of action. And we believe that it is in this context that academic initiatives such as EU-OPENSCREEN, but also others, that aim to develop chemical probes, which are critically important for the validation of novel therapeutic targets, make an important contribution. Because in fact, it's not only industry, but also we observe that academic scientists focus on previously well-studied molecular targets. And you can see here on the graphic on the left, it's very few genes where you see a lot of publications, and the vast majority of the cellular targets are rarely studied. And research in chemical biology can also be rewarding because studies that introduce a novel chemical probe for a cellular target are usually published in high-impact journals and are usually among the most cited publications on that particular target. And we and others observe that if a new chemical probe for a given protein is developed and shared with the scientific community, it strongly encourages other researchers to study this particular protein. And therefore, chemical probes, together with antibodies and protein structures, are arguably the best tools to advance research on a given cellular and therapeutic target, for example, in neglected diseases and in other areas. In order to give you an idea of the size and how these compounds look like, I'm showing here a few examples of, let's say, traditional chemical probes, which have been instrumental in advancing our understanding in many biological areas. And besides their use in basic science, such compounds still represent the majority of marketed drugs today. And in order to support the broader scientific community in developing these valuable compounds, and in order to democratize access to these screening platforms and screening compound collections, which are very expensive to establish and operate, 8 countries joined forces and are currently supporting EU-OPENSCREEN. And EU-OPENSCREEN has been established as a not-for-profit ERIC, European Research Infrastructure Consortium, in 2018, and the headquarters and the central compound management facility is located in Berlin, in Germany. EU-OPENSCREEN was first included in the European S3 roadmap for research infrastructures in 2008, and it is now an S3 landmark project on the current S3 roadmap. EU-OPENSCREEN is a distributed research infrastructure with 24 partner sites across Europe in our 8 member countries. And we basically have 3 types of partner site categories. Mostly it's compound screening platforms and chemistry groups, which together with researchers collaboratively develop novel valuable chemical probes with external scientists. Importantly, the selection and accreditation of these institutions that want to become an EU-OPENSCREEN partner site is based on a thorough 3-step procedure to ensure that the sites on the one hand side have the political support from the national funders to support them on a long-term basis, and on the other side to ensure the scientific excellence of these partner institutions. But how do we now work together with international researchers? Well, we have, let's say, 3 distinct groups of researchers who collaborate with us. First, we have the biologists who have developed a biological assay, but who typically do not have access to these highly automated screening platforms or have no access to larger compound collections, because these are very expensive to purchase, maintain, and operate. So this means a couple of million euros investment by their hosting countries. And the biologists typically contact us when they are interested in developing chemical probes for their own research projects. Second, we have the chemists who may not have the possibility to systematically test their compounds in the different biological contexts. So they contact us so that their compounds can be tested by other biologists in different cellular assays, with the hope that these compounds may have a valuable activity in certain diseases or other applications. And third, we have the database users, because EU-OPENSCREEN, as I mentioned, makes the primary screening data available in our open access database in order to maximize the reuse of these datasets for the benefit of the larger scientific community. And EU-OPENSCREEN is open for researchers from Latin America as well, and in particular, biologists who developed an assay and would like to identify a chemical probe or drug lead for their protein of interest. Through EU-OPENSCREEN, they can use the highly automated screening platforms, they can use our screening collections, or they can receive support from our chemistry experts for the optimization of these compounds. And as these screening projects are usually, let's say, more demanding in terms of resources, consumables, etc., we are also open for discussions about preparing joint grant proposals together with our partner sites. And on the other side, chemists from Latin America can make their compounds available through EU-OPENSCREEN so that they can be tested by other biologists in order to identify valuable novel activities and applications for these compounds. And importantly, the submitting chemists, they continue to own these compounds, and the access to these compounds is regulated, and these compounds can only be tested at one of our partner institutions, and the submitting chemist will be notified whenever these compounds are tested. And we have a model to share novel IP with the submitting chemist. So if you have an essay and are interested in a joint screening project, or if you're interested to hear more about our compound submission model, just drop me an email or send me a message via Uber. I'm happy to organize a bilateral Zoom meeting to discuss your project in more detail. And with this, I close. I thank you very much for your attention, and I'm looking forward to your emails. Thank you very much.